Good evening, everyone. I have one question. How can a 3 pound mass of jelly that you can hold in your palm imagine angles, contemplate the meaning of infinity, and even question in its own place in the cosmos? Especially inspiring is the fact that any single brain, including yours, is made up of atoms, but it requires a lot of protections. So today we are going to learn about such type of protection that is provided by the exosomes. So the role of exosome in neuroscience we are going to learn with our today guest. So please welcome our guest of the day, Dr. Sumedha Mukherjee ma'am. Sumedha ma'am, thank you for joining us today in our session. I hope you are able to hear us.
Yes, I'm hearing.
Yeah.
Thank you,
Thank you so
Karishma.
much, ma'am. Thank you so much, ma'am, for doing this for us. I would like to tell to my all the viewers that Dr. Sumedha Mukherjee ma'am is currently working as an assistant professor in the Faculty of Life Sciences at Jaypee University, Anup Shahar. I hope most of you are from here who have heard the name of this place. Priorly, she has worked with JU and she worked as an assistant professor at the School of Biomedical Sciences at Galgotia University, as well as she holds a strong research background in drug discovery for cerebral ischemia reperfusion injury. I hope most of you are having a research background related to the cerebral part, as well as after completing a PhD in the biomedical engineering from IIT BSU, she moved to Lise, Belgium for her postdoc. So now it's time to take the information from her. So now it's up to you, ma'am. Please start and go ahead. Thank you.
So, is my screen visible?
Yes, ma'am, it's perfectly visible. I request all the viewers to please be attentive and you can open your camera and get ready with the questions throughout the session.
So, one second. So, hello everyone. As Karishma introduced me very nicely, I am Dr. Sumedha Mukherjee, and I'll be talking about exosomes and how they can act as potential tools and potential elements for drug delivery and in neuroprotection. So when we talk about neuroprotection, first thing that we should think about is what we are protecting. From what we are protecting. So we are protecting our neurons, that is the nerve cells, from inflammatory and oxidative damages that occurs to them when there is an incidence of a neurodegenerative disorder. It might be a disease like Alzheimer, it might be a disease like stroke, which can be hemorrhagic or which can be ischemic. Okay, so my presentation today will cover the major topics of the obstacles that we face in central nervous system drug delivery, how exosomes work as a therapeutic agent in stroke model, both ischemic and hemorrhagic, and therapeutic potential of exosomes in another kind of neurodegenerative disorder, that is the Alzheimer disease, and pros and cons of this very recent type of drug delivery. So when we talk about drug delivery into the central nervous system, that is majorly in the brain and spinal cord, we face we face a lot of obstacles actually, because we have come a long way in the cancer research, but when we talk about the neurological disorders, we do not have that much successful drugs in this field also. So what are the major problems that are causing this hurdles in designing a proper drug that can treat our diseases, that can cure our neurological disorders? So the major barrier for a CNS drug delivery is the blood-brain barrier. So the barrier that protects our brain from the blood-borne diseases or blood-borne infections, which stops non-required elements or stops stops the microbes or viruses from entering the brain. That blood-brain barrier itself stops the rescuing drugs to come inside the brain and do their magnificent work, because blood-brain barrier is designed in such a way that it is it is a selectively permeable barrier. So it only allows very few elements inside the brain that the brain actually needs for working, and stops any other kind of particles from entering the brain. The next is the blood cerebrospinal fluid barrier, which acts in a similar kind of way like the blood-brain barrier and stops any kind of unwanted material from the cerebrospinal fluid to enter the brain. And the next is the blood tumor barrier. And if as I was telling about the blood-brain barrier, it is built in a way to protect the microenvironment of the brain, and that is why it is made up of several neuronal cells like astrocytes, neural cells, and microglia, which makes a tight junction that completely restricts the entry of any kind of blood-borne substances into the brain, and thus its main job is to protect the brain. So the question might come that if the blood-brain barrier is doing such an excellent job, then why do we need these kind of drugs to protect the brain, and why are we having diseases at all are that we need these drugs? But the thing is, when there are, which is the main attribute of most of the which are main attributes of most neurological disorders, that is the oxidative stress or the inflammatory responses. This sometimes breaks down or forces the blood-brain barrier to malfunction, and that's when the problem starts. So as we were talking about the blood-brain barrier, what are its characteristics that limit the drug delivery in the CNS? We know this first so that we can have a proper journey of why we need exosomes at all. So, since as I said that it's a very selective barrier, and it will only allow small molecules to enter the brain and restrict any compound that has a molecular weight greater than 400 to 500 kilodaltons. So mostly all the small molecules that we are trying on in labs fails to translate clinically because they are unable to pass through the blood-brain barrier, and hence the molecules that works wonder in the laboratory condition fail spectacularly in the clinical trials when they are studied in a larger population, and thus the very less molecules, like less than 10%, it's a statistic, so they can enter the brain and they can finally work. So thus we have very less amount of drugs which can actually work on this kind of pathogenesis. So obviously the scientists aren't sitting still. We are always working hard, and people around the globe are working hard to find out strategies. The first strategy was our very old friend, the nanoparticles, since these are very nano-sized molecules and very adaptable to be modified with any kind of drugs. So nanoparticles were till in the last decade, it was one of the best bets to go past the blood-brain barrier. Then we also tried to exploit the receptors present on the blood-brain barrier to initiate an antibody-mediated delivery, receptor-mediated antibody delivery to overcome the blood-brain barrier. And since blood-brain barrier allows lipophilic compounds, another idea was to improve the lipophilicity of the drugs so that it can pass through the blood-brain barrier and go into the brain. And the other route was quite direct, which has been used for quite some time now, which was the invasive technique. But this technique has a problem that it needs a very high level of expertise to introduce your drug of interest to the actual place of interest where you want it to go. So any kind of folly or any kind of malfunction, and we are playing with the brain. So this was also not a very good option. But invasive techniques, direct administration to brain, it's still under a lot of research, and it still is one of a very good bets. But then came the idea of delivering drugs using exosomes. So what are exosomes? Exosomes are actually small particles, small extracellular vesicles which are secreted inside our own body. So exosomes are synthesized inside the cells depending on the originator tissue, and it is formed from the endosomes. So when the early endosome forms the late endosome by going through the different steps of forming the intraluminar vesicles, forming the multivesicular bodies, and finally the small particles which are sort of an excreted part of the cell comes out of the cell itself, which are known as extracellular vesicles or EV or exosomes. Sometimes extracellular vesicles, EV and exosome, these two terms are used interchangeably. This small particle, exosome, is actually a big, it has a big caliber. So it has lots of targets around it. It has lots of receptor. It contains lots of transcription factors. It contains a few enzymes. And the most wonderful part about this is it has all the properties of the cell, of the tissue from where it has originated. So, if we need to define the exosomes are tiny vesicles which range from a size of 30 to 100 nanometer. They are found in almost any kind of eukaryotic body fluids, and in our body they mediate lots of important physiological function. And as I said, the exosome always reflects the properties of the donor cells. And since it's produced by almost any kind of cells, they are produced in highly abundant numbers in our body. So how do we design tissue-specific exosomes? So to design a tissue-specific exosomes, what we need is, we need to harvest the hematopoietic stem cells. The hematopoietic stem cells are normally found in the bone marrow, as we all know. So there are certain drugs which are used on the person to help the hematopoietic stem cells mediate from the bone marrow to the bloodstream. This kind of hematopoietic stem cells, which are migrating from the bone marrow to the bloodstream, are known as the mobilized hematopoietic stem cells. Now, this hematopoietic stem cells, or the HSCs, are now easy to harvest because they have now endowed. So they are isolated from the peripheral blood, they are collected and they are differentiated into the dendritic cells, which are the neurons. So we are mimicking since we need to go inside the brain using the exosome, we are differentiating out the hematopoietic stem cells into the dendritic cells, and they can have the characteristics of the dendritic cells, and they produce the exosomes, and these exosomes are collected. You can put your drug of interest. It can be any kind of drug that you want to put in it, and now that the exosomes have their cargo loaded onto it, they are tested for quality control and they can be finally administered. So why are exosomes interesting therapeutic agents? So as you can see in the image here, exosomes have lots of microRNAs, mRNAs of their own. They contain the cytoskeletal proteins and they have a large array of membrane proteins. These membrane proteins help them to bind to the receptors on the blood-brain barrier and pass through it quite easily. And also, exosomes are very flexible. You can use any kind of cargo, that is any kind of drug you want to load into them. Those can be macromolecules or small molecules. You can use double-stranded DNA as a therapeutic agent. Proteins can be used to be loaded into the exosomes, and also single-stranded RNAs. So to understand the effect of exosome in the neurodegeneration and neurodegenerative disorders, I have used a few publications as a model. So first we will look for the therapeutic potential of exosomes in stroke model and in the ischemic stroke model. So as I said before, stroke is a condition where there is a lack of oxygen and nutrient flow inside the brain. So this can happen either due to a blood clot in the brain, in the cerebral artery, where the blood doesn't find its way to move inside the brain capillaries, or it could be hemorrhagic, where a artery breaks open and again there is a lot of blood loss, but the blood is not going into the brain capillaries to give it the necessary oxygen or the nutrient level. So to mimic the actual ischemic stroke, normally the model that is used in lab is the middle carotid artery occlusion or the Macau model. So after this model is based on the idea of closing the sorry this model is based on closing the middle carotid arteries of the experimental animal, and after blocking it for 30 to 40 minutes, the arteries are open. This causes a sudden gush of the blood back into the brain. This particular situation is known as reperfusion. And since there is a sudden gush of blood moving inside the brain cells, the neuronal cells, this causes a sudden oxidative damage. So this model is also known as ischemic reperfusion model. So in this particular study, what was done is that the stroke was induced in a rat model using middle carotid artery occlusion followed by reperfusion of 1 hour. After that, the animals were treated with 10 microliter of exosomes which contained curcumin for 7 days, and then the rats were sacrificed to look for further studies and to find if the strategy has worked. So to monitor cerebral stroke, there are few parameters. The first parameter is cerebral infarction. Cerebral infarction is a situation where the brain cells die due to lack of oxygen. Due to the onset of cerebral stroke or cerebral ischemia, a part of the brain tissue dies off. So that region is almost white when treated with some particular stains, as against to the red color of the stain which is seen in the live tissues. After that, there is another condition which is known as the cerebral edema. This happens due to the breaking down of the blood-brain barrier, which is the third parameter, and due to the breakdown of the blood-brain barrier, what happens is there is a lot of water flows into the brain and there is an increase in the brain water content. If we look into this study, you can see the brain portions. So I will explain the groups. So first one is the sham, which means it's a normal rat which has undergone all the experimental procedures, all the surgical procedures except for the occluding of the middle carotid artery. So as we see in the sham group, there is no cerebral infarction. The whole brain tissue is red. And since the next one is sham and the mesenchymal stem cell derived exosome and the curcumin, there is no change in the first two groups because there was no induction of stroke. But if you look at the third panel, it is the sham and ischemic reperfusion group where there has been a ischemia and a reperfusion. So there is a very clear visible site where there is no red staining. This staining is actually known as a TTC staining, a stain that stains all the living cells red and the dead cells remain white. So you can see in the third panel there is a high volume of cerebral infarction which is indicative of occurrence of ischemic stroke. And in the last panel, where the rats have been treated with the mesenchymal stem cell derived exosomes along with curcumin, you can see that there is a wonderful reduction in the sham, in the infarction. So this particular image confirms the incidence of cerebral stroke that the animal has actually undergone cerebral ischemia. So After that, if we check the water content, we will see similar results as that of the TTC staining, where we saw the cerebral infarction being increased in the IR group and reduced in the treated group. But also you have to check the reactive oxygen species. As I said that reactive oxygen species, uh, as I said that the most of the neurodegenerative diseases are characterized by oxidative stress, and oxidative stress is mediated by the presence of reactive oxygen species. When there is an increase in the oxidative stress, which is a main characteristic of any kind of neurodegenerative disorder, there is an increase in the percentage of the ROS or the reactive oxygen species. So as we see here, there is an increase in the ROS percentage in the ischemia reperfusion model, which is reduced by the treatment of the exosomes. So this picture is a immunofluorescence study. This is a immunofluorescence staining. So here also we have exactly the same groups as we have seen in the previous slides. So here let me explain the molecules a little bit, which are the markers here. So first panel, the green one, you see it's a ICAM. So ICAM is intercellular adhesion molecule. This is a marker of neuroinflammation. So when there is an increase in inflammation or increase in damage in the blood-brain barrier or in the brain, this molecule has been shown, this molecule acts as a signal. So if there is a higher expression of ICAM, we can say that there is a impending damage on the brain tissue. There is an inflammation in the brain cells. Similarly, with the V-cadherin, which are also adhesion molecules, we can see that in V-cadherin is another marker which is normally reduced in case of neuroinflammation. So if we look at the first two panels, the sham and the sham plus exosomes, these two panels, there are not much changes in the levels of ICAM or V-cadherin. But if we look at the lower panels, both of the lower panels, you will see that ICAM has intensity of ICAM, the green fluorescence has increased a lot as compared to the first two panels, and it again gets reduced when it is treated with the exosomes. And if there is a problem in understanding the image color, the fluorescence is quantified and presented in the graph below, where we can see the same thing. The green column is for the ICAM and the red one is for V-cadherin, where we can see there is an increase in the ICAM percentage in ICAM intensity in IR group, and there is a reduction of the V-cadherin intensity in the IR group, which are both alleviated by treatment with the curcumin loaded exosomes. Similarly, there are two more markers which has been tested, chlorine 5 and occludin, and we have similar results here. These kind of results that the exosome has been able to deliver the drug curcumin inside the blood-brain barrier, and where it has started working, and we can see those results visually. To move on to the next type of model, which is hemorrhagic spoke model. This was also performed in a animal model, and the hemorrhagic model was induced by a blood injection, and the MSC derived exosomes were injected in the body using a tailwind injection. So like ischemic stroke has a markers in cerebral infarction or blood-brain barrier damage or brain water content. What we see here is, in the case of hemorrhagic stroke, there are changes in the spatial memory. So in this study, the scientists have looked into the memory tests for almost over a month, and it was seen that while the spatial memory has decreased in the hemorrhagic or the test group, there was an increase in the in the spatial memory and recognition learning function in the animals which receive the treatment of curcumin loaded exosomes. Also, any kind of hemorrhagic stroke or any kind of stroke, brain stroke actually causes a sensory motor deficit. So the sensory motor deficit was also checked. While initially there was no significant results, over a longer period of treatment, it was observed that the sensory motor function of the treated animals were much better as compared to the control group, which is the rats that suffered from hemorrhagic stroke. So, stroke being one of the most severe of the neurological disorders, which still do not have any kind of treatment as we have seen till date. It has only one drug, one FDA approved drug, which is the RTPA, recombinant tissue plasminogen activator. So these kind of studies, this kind of results are very important for us because these gives us hope of future drugs in neuroprotection, which gives us hope that there might be someday we will develop a drug which will protect the humans from the trauma of cerebral ischemia or hemorrhagic stroke. So, moving on from the stroke, there is another very well-known neurodegenerative disorder, which is Alzheimer's disease, which is a age-based disease, we can say, or which is a disease different than that of the stroke. This disease is characterized with loss of cognition, uh, sensory deficits, and loss of memory, and brain dysfunction, a sort of memory loss in the brain. So Alzheimer's disease models, it is very difficult to induce Alzheimer's using chemicals. There has been lot of trials, but the transgenic models are quite close to the Alzheimer pathology that we see in humans. So this is a APP-PS1 transgenic mice which highly resembles the pathologies of Alzheimer's disease, and the effect of exosomes were tested on this model. Just like we have discussed the characteristics of stroke pathophysiology, we will first discuss the pathophysiology of Alzheimer's disease. So the main attribute of Alzheimer's disease is formation of amyloid beta plaques. There are deposition of amyloid beta between the neurons, which inhibits the signal transmission between the neurons. Then there are the downregulation of the amyloid beta degrading enzymes. So there are there are lots of enzymes, but there are few enzymes that are responsible for degrading this amyloid beta plaques that are getting deposited in the brain. So if these enzymes are working properly, they will be able to degrade the amyloid beta plaques, and there will be a hope that this delay the onset of the symptoms of Alzheimer. So one is NEP or neprilysin, and the other is IDE. So IDE is insulin degrading enzymes. So this is also known to have a very interesting function in degrading the amyloid beta deposition. So if these two enzymes are downregulated, as we can understand, they will not be doing their functions properly, and the aggregated amyloid beta plaques will stay wherever they are. So another one is activation of the microglia in brain. So accumulation of amyloid beta fibrils or amyloid beta plaques cause activation of the microglia in the brain, which is a signal of the neuroinflammation of brain. So let's look at this image. So here we can see the hippocampus of the brain and the cortex of the brain. So the control group here is the APP-PS1 mice, which is a model organism for Alzheimer. So the control group itself is the diseased, is the mice with disease pathology. So when we, so if you look at the bright dots, bright green dots, they are the, they are the amyloid beta deposition. They have been stained with a particular dye known as thioflavin S, which gives it this green, frozen green color. So you can see that in both hippocampus and cortex region, in the control group, in the epipps1 mice, there is a deposition, high deposition of amyloid beta. And if we look at the MSC exosomes treated, it's just only exosomes. So if we look at the mesenchymal cell derived exosomes treated mice, you will see that there are lots of less number of amyloid beta plaques. There is a reduction in the amyloid beta deposition in both hippocampus and cortex. This helps us understand that the exosome, it has shown some effect in regulating the deposition of amyloid beta. So it not only the exosome treatment has reduced the deposition of amyloid beta, but it was also shown that the treatment with the MSC derived exosomes increased the expression of the enzyme NEP and IDE. So this is a Western blot. If you, I think most of you will be well versed with it. So in the control group, you can see that there is a reduction in the expression of the both enzymes, and after in the mice group where the animals are treated with the MSC derived exosomes, so there is an increase in this, in the levels of this proteins again. So increase in the levels is a good indication that they will be able to break down the amyloid beta plaques. So we have discussed the two major factors. This image represents the microglia activation. So microglia active marker for microglia is the IBA1. So this is the immunofluorescence image again, where there are antibodies used against the IBA protein, which is a marker of microglial activation, and we can see that in the control group, and there is a excessive, you can see the red dots. Those are the anti-IBA antibodies. So these antibodies have bound to the IBA, and this indicates the activation of microglia in the cortex and hippocampus of the Alzheimer pathology mice. And in the other group, where there is, the mice have been treated with the MSC exosomes, we can see that there is a reduction in the immunofluorescence correlating to the reduction of the microglial expression. Also, since there is a neuroinflammation, there is always role for the pro-inflammatory cytokines. It was observed that in treating the, after treating the mice with the exosomes, there was a reduction in the expression of the pro-inflammatory cytokines such as IL-1 beta, TNF alpha, TGF beta and IL-10. So what are the beneficial aspects? So using these three different models of three different diseases, we can safely say that exosomes really hold a great potential in treating the neurological disorders, and definitely it can pass through the through the blood-brain barrier, and it can reach the brain, and it can work its magic. It can deliver whatever cargo you have given to it. So what are the beneficial aspects of the exosomes in neurodegeneration? So they can provide neuroprotection. They have shown improvement. The treatment have shown improvement in the behavioral changes. They have been able to successfully reduce the inflammation. They have shown immunomodulation, and in case of Alzheimer's disease, they have also shown reduction of plaque formation. Also, other studies report that they can help in neurogenesis, synapse formation, and also in white matter remodeling. So, like every good therapy, there are some pros and there are some cons. So, the pros of the exosome therapy are they are quite effective because they can successfully deliver any kind of drug or any kind of material, any kind of therapeutic material for fact into the brain. They can carry any kind of therapeutic material, be it DNA, be it RNA, be it a protein, or be it any kind of phytochemical. So it can carry anything. So they are biocompatible, of course. They are a part of they are normally synthesized in our body, so are highly biocompatible. They are highly specific to the target cells, having all those kind of molecular markers. They are permeable through the blood-brain barrier. They have low toxicity, and the most important thing, they induce low immune reaction. They have a very low immunogenicity. The cons are that this is a problem till date. There is a isolation of exosomes still remain a big problem. There are those protocols need to be standardized. They need to be scaled. So one of the main con is the isolation method itself, and also optimization and efficient drug loading into the exosome. Those approaches still need to be worked on a little bit, not little bit, quite a bit. So, and also exosomes cannot be stored for a very long time. They doesn't remain stable. So their long-term storage stability still remains an issue. For listening to me, and I will be open to take any kind of questions that you have, but I am running a little short on time, so if you could, if anybody have any kind of question, I will be happy to answer.
Participants, you can start asking your questions if you have any doubts.
Yeah, all the participant, I request to please put your question. I have one question, ma'am. Like,
Yes, sure,
as
Karish.
you've done the really, yeah, as you've done really a wonderful research over it. So I just wanted to know what are the challenges that you have faced while working on these exosomes?
As I said in the last slide, that the biggest challenge in exosome therapy is to isolate the exosome properly. Like as I said in the slides, that the idea to derive an exosome for neurological neurotherapeutics, if you want to deliver a neurotherapeutics into the brain, so you have to sorry, so you have to derive the exosome from the any kind of stem cells. So you have to derive the stem cell, which is itself a challenge, and then you have to get the exosomes isolated from the blood. So what we really need is a standardized isolation protocols and standardized isolation methods for exosomes, so that it becomes highly scalable. Because when we are working in labs, we can do trial and error and everything, but when it comes to the clinical trials or it has to go to the pharmaceutical market, you really need proper scalable methods, proper reproducible methods where you can justify your procedure, right? So that remains one of the biggest challenge to properly isolate and scale the exosome isolation.
Thank you so much, ma'am, for this wonderful answer. There is one more question in the queue that is asked by Imtiaz. What are the storage conditions like for transportation of exosomes?
Is it in the chat? Because I
Yes,
missed.
ma'am. There are two questions in the chat in the
Okay,
queue.
one second, please. So Um, Shelf life in exos. I see two questions. Yeah, both are the similar questions. So exosomes in 4-degree in lab conditions can be stored for like 5 days, mostly a week, and in -20 they can be stored up to like 3 to 4 months. So as you can see that this could be a really big problem for using them for long term treatments. So this is okay for working in lab, but to get it to a clinical trial level and to use them as drugs, these storage conditions really need to be improved. Anything else?
Ma'am,
Imtiaz and Suresh, did it was it okay? Did it satisfy your query?
yes, ma'am. Another query is like, what is the media that we can use to store this? Because of the shorter shelf life. So, any specific media you recommend to use for storing?
I will. mm, So this is not about the media actually. So this is mainly about, since they are the, sorry. So if you, I don't know if you are aware of, but there are some particular kits that are used for the exosome isolation, and you can use any kind of normal cell culture media actually for the exosome uh, for storing the exosomes, because even when you are doing cell cultures, if you take the supernatants, you will find some exosomes in it, because basically they are extracellular vesicles, so they are secreted by any kind of cells. So there are some particular companies, and there is one startup in India also which is known as Xsure. So Xsure is working tirelessly on making up kits and reagents to help you with your all kind of exosome needs. So they are using they are trying to make up reagents, and they are trying to make up some kind of they're coming up with some ideas, and they have some kits I saw on their website. So they are trying to make up some better management in exosome therapy, exosome isolation. So I think it will, if you are really interested in how to manage the exosomes and isolation and all the things, you should really go through their website. It's known as X. I will put it in the message. You can go through their website, you will see that they are working on a lot of stuff regarding the exosome.
Thank you, ma'am, for this wonderful session.